Welcome to our final unit of the year. We are going to spend the time remaining to us in this course looking into how things grow and develop and why living things look and inherit the traits that they've got. And that study starts with looking into our final biomolecule. I promised you that we would get more information about nucleic acids. This week is when we're going to do that. So, this week is all about DNA, RNA, and how do those things work? How do we make more of them? By the end of this lesson, we will have gone over how do you start with one strand of DNA and make more copies of it? And how do you go from DNA to protein? So, just to refresh your memory, DNA is one of our four biomolecules. It is located within the nucleus of cells, and it is found in this form that we call chromosomes. So, a chromosome is just a tightly coiled, organized bundle of DNA. DNA controls what proteins the cell makes and what versions of those proteins, if there are different options, it can make. And to remind you, proteins then are the major structural unit of organisms and control all of the chemical processes within the cell. And to remind you, DNA stands for deoxyribonucleic acid. So here's a little closer look at DNA. And it looks sort of like if you started with a ladder and then gave it a twist. So we call this structure a double helix. The side rails of the ladder, the vertical parts, are made up of phosphates and sugar molecules. Specifically in DNA, that sugar is called deoxyribose. And then the rungs of the ladder are nitrogenous bases. There are four different types of them, and they are held together. So there's one on each side rail, and they're held together in the middle by hydrogen bonds. RNA is very similar, but instead of being a double helix, it's usually found as a single strand. So it's got its sugar phosphate backbone, a different sugar, ribose instead of deoxyribose, but otherwise the same kind of structure, with these nitrogenous bases attached to it. Let's take a closer look at those base pairs. And they look a little bit like the carbon rings that we talked about when we talked about sugars, except that there's a lot more nitrogen in them than your typical sugar would have. And there are two of them, thymine and cytosine, that are made with a single carbon ring, and two of them, adenine and guanine, that are made with a double ring. Notice in this diagram that they come in pairs because there are natural places where hydrogen bonds want to form. Adenine and thymine have two of those places between the hydrogen at the top and the oxygen, and between nitrogen and hydrogen down below. Cytosine and guanine have three spots that want to form hydrogen bonds. So they match up perfectly. And what that means is when you start forming a molecule of DNA, if you have adenine on this side, it will always be attached to thymine on this side. And if you have thymine over here, it'll always be attached to adenine. Same with guanine and cytosine. If you have one of them on this side, you will always have the other one on this side. And that's very important because that starts to allow us to build a code that is going to control what an organism is able to make out of protein. So we can break DNA down into the sections that code for different proteins. And we call those sections genes. You've likely heard that word before. A gene is just a segment of DNA that codes for a specific protein. And then the protein that is made results in another word that you're probably familiar with, a trait. We're going to get now into some of the specifics of how DNA works. These are the basics about DNA that you will need to understand in order to get to the fun stuff like cloning and genetic disorders and reproduction. So, in order to get to those things, we have to master a couple of concepts. We have to master how DNA works, how it does the job of creating protein, or is involved in the job, I guess I should say. And we have to know how DNA makes more DNA. So we'll start with the function of DNA. And DNA works in its job of helping your body make protein in 2 steps. Step 1, DNA can't leave the nucleus. So we've got to have a way to essentially make a photocopy of it so that we have something that can leave the nucleus and get to the protein making machinery. And then step 2, the protein making machinery works with that photocopy to actually make a protein. This is all on your screen in the more formal language, but those are the basics. So that first step, making a photocopy, the word for that is transcription. And this happens because DNA can't fit through the pores that let things in and out of the nucleus, nor do we really want it to, because you really want to keep the DNA somewhere safe, somewhere where you're going to be able to use it over and over again to make new proteins. If it's just out floating around in the cell where any old thing could run into it and damage it, that's less than ideal. So we keep the original safe inside of the nucleus, and we make photocopies that we can send out to work with. Those photocopies are called messenger RNA or mRNA. So we unzip the DNA, we pull the latter apart, and then a protein can come through and attach new bases to each of the bases on one side of the DNA. So in that way, it is making a copy. Because remember, if I've got adenine on this side, I'm always going to have thymine on this side. So by lining those two up, I can make sure that I have the exact same information all the way down. There's one little change in RNA, though, because RNA doesn't have thymine. RNA still has 4 base pairs. Cytosine and guanine and adenine stay exactly the same, but in mRNA, instead of having thymine, we have a 5th nucleic acid called uracil. It fills the exact same spot. It works very similarly. It's just a slightly different structure. So we've taken our DNA, we've pulled it apart, we've made a photocopy. That photocopy travels out of the pores of the nucleus into the cytoplasm. And it floats around in the cytoplasm until it bumps into a ribosome. Remember, ribosomes are our protein-making machinery. Ribosomes then go on to read the mRNA. They read groups of 3 bases to make 1 amino acid. This seems unnecessarily complicated, but there's an important reason for it. Let me show you. All right, check it out. So we have 4 nucleotides, right? We've got adenine, thymine or uracil, guanine, and cytosine. We have to get to about 20 amino acids. The number is a little variable, but 20 is a good ballpark. Let's say that we read these one at a time. So A could code for a single amino acid, T could code for 1, G could code for 1, C could code for 1. Obviously, that's not going to work. That's only getting us to 4 possible amino acids, right? When we add those up, that's nowhere near enough. So we've got to read them in combination in order to have enough that we can get the work done. So let's clear that out a little. And let's say, what if instead of reading them one at a time, we read them in groups of 2? So we read adenine, or we could have a group of AT, or a group of AG, or a group of AC, or TA, TT, TG, TC. See where I'm going with this? So with this one, if I fill it all the way in here, now we have a situation where if each one of these combinations codes for an amino acid, we can have 16 total amino acids. And that's better, but it's still not enough. We still need to have more because we know that there are around 20 of them in all. So to get to that magic number of 20, we have to read amino acids in groups of 3. So we have to have 3 of them in total together. And I'm not going to write this all out because it would get ridiculous. We're just going to do a little math instead. So we started with, if we were reading them as singlets, 4 possibilities, multiplied by 4 to get to our reading them as doubles. We multiply by 4 again to read them as triples for a total of 64 possible combinations. And that means that we can have a unique 3-letter nucleotide code for every amino acid. And in fact, we can have it so that many of the amino acids have 2 or 3 3-letter codes that will produce that amino acid. So using that 3-letter nucleotide code, the ribosome can read the mRNA and use another kind of RNA, tRNA, to start building a protein where every amino acid has a 3-letter nucleotide code that represents it. So in this diagram, you can see a better view of what's actually going on during that second part of the process of making a protein, during that translation phase. And what you see here is that the mRNA gets fed through the ribosome. And as it goes through, these pieces of tRNA each have a little snippet of nucleotides on the end that correspond to the ones that are getting fed through on the mRNA. So as you read across this one right here, you see that the original mRNA has UGC as the first 3 pairs in the sequence. And so that U matches up to an A. That would be its complement because adenine and uracil attach together just like adenine and thymine do in DNA. Cytosine attaches to guanine, and guanine attaches to cytosine. And that 3-letter code codes for the amino acid alanine. The next 3 letters, AGA, code for the amino acid that you see next in the list. The next 3 correspond to the next amino acid. So tRNA brings in those amino acids, and the ribosome links them together to form a polypeptide, which will get folded into a protein. That's the first part in a nutshell. So far we've done going from DNA to protein. The other process that you need to understand before we can move on to the interesting stuff and the fun stuff is occasionally we need to make more DNA. And it would be really bad to just glue some nucleotides together in a random order and hope for the best. So there is another process that's going to look very similar to transcription. You're going to see some of the same stuff happening, but its purpose is to start with one DNA double helix and make from that 2 perfect copies of the original. The process is called DNA replication, which, makes sense. To replicate something means to make more of it. Here's how it works. So we are going to, just like with our process of transcription, unzip that DNA. So we're going to pull apart the 2 strands using this protein that we call DNA helicase. There's another protein in there, primase, that's going to get the DNA ready to copy. And then proteins called DNA polymerases can add on to the existing two sides of the original DNA new nucleotides. So that, in a nutshell, you start with one piece of DNA, you pull it apart, you add new nucleotides to each side, and you end up with two new pieces of DNA, each one of which is made up of half of the original and half brand new nucleotides that have been attached to it. There's some other details to this. Like you'll notice in this diagram that we have a leading strand and a lagging strand. And what that relates to is that DNA polymerase only works in one direction. Meaning, remember we talked about the fact that the sides of the ladder are made from this sugar phosphate pattern? Well, those sugars, they're directional, meaning they point in one direction on this side and in the other direction on this side. I'm going to put in a diagram in a minute to show you what I mean. So if that doesn't make sense to you yet, hang tight. But DNA polymerase can only run in one direction. So on the leading strand, it's working just fine because it's going in the direction that the sugars are pointing. But on the lagging strand, the sugars point the other way. So as it's unzipping, that DNA polymerase has to keep jumping and then filling in with some nucleotides and then jumping back up to where the DNA has been unzipped and filling in some nucleotides, which results in little fragments that then have to be glued together. So let me show you a diagram to explain a little bit more of what I mean here. So here is a zoomed in look at DNA. And just to make a notation, this is the official moment where I am not going to ask you to memorize this. This is strictly for the sake of clarity and for the sake of those of you that are going to go on to AP Bio where you might be asked to know this. So what you're seeing here, let's switch colors so that I can make it stand out. Right here is a single nucleotide. And this right here, this is your phosphate. This right here is your sugar. And this here is the base. Notice that on this side, all of the sugars have a point that is pointing that way. And on this side, all of the sugars are pointing the opposite direction. Now, in organic chemistry, we name the carbons in that sugar ring, and we are highly creative. We name those carbons 1, 2, 3, 4, and 5. I know, incredibly complex. So we start counting the carbons from the one that's attached to the base. So this one here is 1, and we go around the circle, 2, 3, that's a 3, I promise, 4, and 5 is at the top. And so when we start to talk about DNA and how it's set up, we refer to the 3' and the 5' end of the DNA. And what that means is down at this end, the number 3 carbon is on the bottom. And up at this end, the number 5 carbon is on the end. So when you start to make new DNA from the old DNA, DNA polymerase can only travel from 3' to 5'. So if you start splitting open the DNA down here, this piece starts to peel off to the left, this piece starts to peel off to the right. The DNA polymerase that's over here on the left side, it's in great shape because it can just jump in and start zooming up the DNA that way because it can travel from 3' to 5'. But if a DNA polymerase tries to jump in on the right side, it can't go that way. That doesn't work. It has to wait until this has split open a little bit, start here, and travel down to the end. And by that point, it will have split open a little more, so it can jump up here and travel down to where it left off last time, and so on and so forth. So this side would be the leading strand because the DNA polymerase can just jump on at the start and zoom all the way until it gets to the end of that DNA without stopping. This side would be the lagging strand because it's constantly having to go down and then jump back and then go down and then jump back and then go down over and over and over again, creating a whole bunch of teeny tiny fragments that we then have to glue together. We're going to wait for the little bar to go away. We're going to flash our hands, then we're going to talk. So the last thing that I want to hit on today is the fact that, like most of being alive, this process isn't perfect. Sometimes things go wrong. DNA polymerase is capable of screwing up the process of making new DNA out of old DNA. And there are safeguards in place to catch those mistakes, but they don't always work. And when something goes wrong and a base pair gets put in a way that it shouldn't have been, what results is a mutation. No, we are not talking X-Men. We're not talking Spider-Man. There is no radioactive spider that bites you and then your DNA changes and then you get superpowers. But we are talking about changes to DNA. And those changes tend to come in 3 forms. The simplest one is DNA polymerase is chugging along. It gets to a thymine. It's supposed to put in an adenine. It screws up and puts in a cytosine instead. And as the editor comes along, it takes a look and says, those don't match, and replaces that adenine that was supposed to be there with the guanine that matches that cytosine. So now one base pair has replaced another base pair in the overall sequence of DNA. The total number of base pairs has stayed the same, but one of them has been changed from what it used to be to something new. This is a substitution. The other two are just as straightforward. You can delete one or more base pairs. They just get left out by accident. Or you could insert some extra base pairs that weren't supposed to be there. All of these kinds of mutations can have minor or profound effects on the final protein that ends up getting produced. Check it out. So let's look at those substitutions, which are also known as point mutations. So here on the end, you see what was supposed to happen. So we had thymine, cytosine at the DNA level. That copies to adenine, guanine when we make the mRNA, which is code for the amino acid lysine. Sometimes a point mutation can be silent, which means let's swap that cytosine out for a thymine, which means that now the mRNA reads adenine, adenine, but that happens to also code for lysine. So while a change has been made at the DNA level, no change is made at the protein level. Or you can make a nonsense mutation, like the fact that there is an amino acid sequence or a nucleotide sequence, I apologize for my misspeaking, that tells the DNA to stop, that says don't add any more amino acids. Or you can have what we call a missense mutation, which is when you end up with a 3-letter code that codes for a different amino acid. And we can break those up into, well, if it's an amino acid that behaves in the same basic way, you might be okay. You might still end up with a functional protein. Or non-conservative, where you end up putting in an amino acid that has completely different chemistry, that behaves in a very different way. And these non-conservative missense kinds of mutations and these nonsense kinds of mutations can have some very drastic effects on organisms. For example, sickle cell anemia, Tay-Sachs, and cystic fibrosis are all genetic disorders that come from single point mutations. So you've probably heard of sickle cell anemia. It's a single point mutation in the gene that creates hemoglobin, which is the protein in your blood that carries oxygen. So what happens with sickle cell anemia is that red blood cells can't carry oxygen properly anymore. Tay-Sachs disease is involved with multiple point mutations in the hexa gene that causes nerve cell deterioration over time. And I'm sure that most, if not all of you, have heard of cystic fibrosis. A single amino acid gets deleted, which creates an improperly folded protein that results in the production of thicker than normal mucus that does things like blocking the airways and impacting the function of the pancreas. All right. I know that that was a lot of information, and normally I would have broken it down just a little bit more, but welcome to the world where nothing is normal. So this week, you're going to practice those concepts of transcription, translation, and DNA replication so that we can move on to talk about some of those things related to mutations and why it is that different organisms look and act different from each other, and all of the cool stuff that's involved in genetics and heredity. So go do good work, be good, be safe, be kind, don't do anything I wouldn't do, wash your hands, and if you have any questions, make sure that you get in touch with me quickly, because this knowledge is going to be the foundation that we are going to build upon for the rest of the school year.